for joining us for today's webinar, Primary Cells 101, presented by AllCells. My name is Afshin Shirazi, and Marketing Manager at AllCells, and I'll be moderating today's presentation. Before we get started, please be aware that everyone has joined today's program automatically muted, and in order to participate in the question and answer session immediately following the presentation, please enter your questions at any time using the GoToWebinar prompt at the right of your screen, and we will address them at the conclusion of today's webinar. We're excited to introduce our presenter for today's program. Joining us from my team, we have Erin Kelly, Bioservices Supervisor. Erin has been with AllCells for 2 years and heads up our Bioservices Department. She has a PhD in Immunology from the Weill Cornell Medical School and has worked with many different primary cells, including monocytes, macrophages, T cells, lipoaspirate, bone marrow, and blood. Please join me in welcoming Erin.
Thank you, Afshin. So in this webinar, we will hope to engage you in a discussion of primary cells and explain to you what primary cells are, the differences between primary cells and cell lines. We will give you many examples of how primary cells are used in today's application. And finally, we will explain how to work with primary cells. And at the end, if you have any questions, I will be happy to answer them. Since the late 1800s, biological research has utilized the cell, newly established as a structural and functional unit of life, as an ethical and workable model for the physiological and biological nature of the organism. In 1885, Wilhelm Roux established the principles of tissue culture by culturing the medullary plate of an embryonic chicken in warm saline solution for several days, attempting to demonstrate Darwinian principles at the cellular level. Sterility and reproducibility of the cell culture method were improved upon over the following century, permitting medical breakthroughs such as the polio vaccine and spurring the field of regenerative medicine with the successful propagation of embryonic stem cells in culture. Later on, cell culture systems of different sorts were put to use in accelerating biotechnology breakthroughs such as recombinant DNA technology, and were basically used as factories for producing human proteins with wide-ranging applications. By mid-century, while researchers were struggling to verify the cellular determinants of heredity, they needed a model that could be reproduced across laboratories. The need for greater supply and consistency in cell models spurred the creation and popularity of the HeLa cell line, a subpopulation of cells isolated from a cervical cancer biopsy that demonstrated abnormal growth properties, specifically to the surprise of researchers, seemingly unlimited growth potential. Other cell lines derived from tumor cells were soon established and used to study not only cell cancer growth, but normal biological processes as well. Over 10,000 patents and 100,000 publications use HeLa cells as a model for the basis of their findings. HeLa cells are one example of what is known as a cell line, oftentimes called a continuous cell line. I'm guessing that many of you in the audience have used a cell line at one time or another, and maybe one day while you were mindlessly passaging them, wondered where the cells came from or how many times they had been passaged. Typically, a cell line is derived from a solid or blood tumor of a patient. The tumor is dissociated into a cell suspension and put into culture with different growth mediums. If the culture is slow growing, the researcher may try to enhance its growth by transforming the cells with a virus, often the EBV virus. If the culture continues to propagate, the researcher will continue to subculture it, intentionally or unintentionally selecting for the fastest growing cells. At any point and at any level of confluence, the cells can be frozen for storage, shipped to collaborators, and thawed for another round of subculturing in a different lab. The end user often has no knowledge of the subculture history. Many scientists have noted potential limitations of conclusions made from such a highly manipulated system and have advocated for the use of a more relevant model. Primary cells are an alternative to cell lines and refer to cells taken directly from living tissue of an organism, healthy or diseased. Primary cells can be taken from a biopsy or more frequently from blood and bone marrow tissue. Primary cells are taken from blood and bone marrow are often referred to as primary hematopoietic cells and are the most commonly used primary cells as they are the easiest to obtain. When establishing a model to test a drug or a biological hypothesis, a logical consideration is its biological relevance to the in vivo system you are modeling. Biological relevance can include such parameters as species, genetics, cell context, and physiology, but ultimately means that your assay readout accurately represents what would occur in the whole organism, organ, or cell being studied. cell lines, by definition, are a homogeneous model, selected over time for identical characteristics of fast division and survival. In contrast, peripheral blood mononuclear cells, or PBMNCs, are a commonly used primary cell that reflects the heterogeneity of the blood, containing lymphocytes, monocytes, and granulocytes. PBMCs, in contrast to a cell line, represent the multicellular complex content of a tissue, in which cell-cell interactions and tissue environment can play a role in responses to external stimuli. Molecular and cellular techniques made available in the past few decades have verified long-lasting concerns of the representative nature of cell lines by revealing tremendous genetic alterations accrued over decades of subculture. Such cell lines show many alterations from the primary cultures, including changes in morphology, chromosomal variation, and changes in functional assays. Cell lines such as HeLa have become so genetically distinct from the human genome that some scientists have proposed that cell lines such as HeLa be given a designation of species. In contrast, primary cells undergoing only 1 to 4 passages outside of the organism reflect an intact genome. Cell lines have come under additional scrutiny lately due to false identity. Published studies have revealed that more often than not, cell lines are misidentified or mislabeled. The scientific publishing house Nature just announced last month that as of May 1st, it would require authors to answer questions about the identity of any cell lines used in the article submitted to their journals for publication. This leads us to think about the important but often avoided topic of variability, which is related but distinct from the issue of heterogeneity. Primary cells, by definition, will have much greater variability from lot to lot or human to human than 2 different vials of the same cell line. This is a challenge for many researchers looking to reproduce results from experiment to experiment. However, researchers must face this challenge head-on, acknowledge the importance of human-to-human variability, and try to understand how these differences affect their research projects and hypotheses. AllCells can help you in this process by supplying primary cells with donors in specific demographics, with specific genetic backgrounds, including HLA typing, and can even do custom screens of donor panels for your projects. Furthermore, if you want to work consistently with a single donor cell to control variability in your project, AllCells can bank the cells for you or recall a specific donor for repeated use. In the past, researchers resorted to using cell lines due to the lack of availability or accessibility of primary cells. Here at AllCells, we can provide immediate access to numerous types of human, non-human, primate, rat, and mouse, hematopoietic, stromal, and tissue-derived primary cells. They can be shipped to you overnight, fresh, or sent frozen. When trying to access primary cells on their own, researchers often worry about the viability and purity of the primary cell population. All of AllCells cells are guaranteed for viability and purity of the primary cell populations. Contrary to some claims, primary cells don't require special media additives and can be maintained in a 10% serum nutrient media. Although not always the case, primary cells can be a higher cost than cell lines, especially since researchers often get cell lines for free from collaborators and can grow them indefinitely. In contrast, primary cells have a limited lifespan and may survive for 2 days to 3 weeks depending on the cell type, and some cells can be passages multiple times. All of this is not to say that cell lines do not have a role in biological research. Cell lines such as HeLa have clearly demonstrated their historical importance, serving as a first-generation cell culture model, contributing to major historical, biomedical, and engineering breakthroughs, such as vaccines, recombinant DNA, and protein production. Due to their growth characteristics, they have been critical in elucidating the mechanisms of self-renewal, cellular transformation, and even oncogenesis. However, the wide gaps that still exist in our understanding of cancer and other diseases may in fact be evident itself of the limitations of cell lines in certain applications. While cell lines may continue to be useful in industrial applications such as the manufacture of animal-derived DNA, proteins, and drugs, the biomedical and disease-driven applications of the 21st century call for more individualized and biologically relevant models. Primary cells will continue to be a standard model, in vitro model, for applications such as drug development, biomarker identification, and the development of cellular and tissue therapies. So these disease and therapeutic-based applications include biomarkers, drug screening, immunotherapeutics, and tissue engineering. Currently, they are multi-billion-dollar industries projected to grow substantially over the next decade. Firstly, biomarker research relies heavily on primary cells for identifying specific molecules or proteins indicative of various pathological states or drug resistance. Take the example of autoimmune diseases treated with immunomodulatory drugs. A group of researchers at Celgene have identified a protein, cereblon, that corresponds with increased drug resistance to immunomodulatory therapies of multiple myeloma patients. In this study, these researchers first generated a multiple myeloma cell line resistance to a commercial cereblon antibody. Then the researchers probed cereblon mRNA and whole protein levels via Western blot and PCR techniques and determined that increased drug resistance correlated with lower levels of cereblon. This research is impactful because it could change the course of treatment for many struggling multiple myeloma patients and lead to the development of a clinically available test for resistance. Featured in this research paper was AllCells CD138 positive purified cells from multiple myeloma bone marrow. In addition to the disease diagnostics, a major field of medical research obviously involves disease treatment and management. In the drug development field, scientists are engaged in identifying and characterizing novel targets for the treatment of major diseases. In particular, screening is a major part of the whole development process and often involves high throughput and long-term testing of therapeutic candidates in cell-based in vitro assays to verify their efficacy and specificity. For example, researchers at Adheron and UC Berkeley use DNA-coded microarrays to immobilize lymphoma cells and then expose the cells to alpha CD20 and measure the drug potency. The researchers built and tested these microdevices with fluorescent labeled cancer cells mixed into PBMNCs, essentially mimicking a whole blood sample. They were able to clearly visualize the immobilized labeled cancer cells and miss the PBMNCs and detect if they were alive or dead. Featured in this research was AllCells peripheral blood mononuclear cells. Within the drug development field, there are many types of models used to test drug potency. One particular type of treatment, immunotherapy, is emerging as a new gold standard in treating cancers, calming the immune response and suppressing HIV. Within these fields, primary cells are consistently used both to test the immunodrug efficacy, similar to described prior, but also to model the mechanisms of the underlying immunological malignancies. For example, researchers at the University of Texas Medical School demonstrated the implications of hyaluronic acid expression in mesenchymal stem cells, and that it plays a major role in MSC leukocyte adhesion, crosstalk, and inflammation. On the other hand, researchers at California Institute of Technology produced large amounts of powerful anti-HIV antibodies in vitro from cord blood CD34 positive cells differentiated to specialized B cells. Featured in this research articles were AllCells bone marrow and cord blood derived CD34 positive hematopoietic progenitors. A final application of primary cells is in tissue engineering, which is part of a growing field of regenerative medicine that heavily utilizes primary cells to regrow damaged or diseased tissue. In a study at Columbia University Medical Center, mesenchymal stem cells, or MSCs, and hematopoietic progenitors were co-cultured on tissue scaffolds and then implanted into mice where enhanced angiogenesis was observed. In scaffolds implanted with MSCs alone, little vascularization was observed. Featured in this paper was AllCells bone marrow and CD34 positive progenitors derived from bone marrow. It is evident how scientists work with primary cells to achieve various research goals. Therefore, we will now briefly discuss how primary cells are isolated and handled, and provide an overview of the downstream applications and processing of these cells. Firstly, at AllCells, cell populations are purified via immunomagnetic separation techniques and also whole tissue isolation protocols. In our industry, Miltenyi, Automax, and Clinimax are regularly used to purify hematopoietic cell populations from peripheral blood, mononuclear cells, and bone marrow. Additionally, whole tissue isolation is used to target primary cell types not found in the blood and lymphatic systems, such as endothelial cells, epithelial cells, adipose stem cells, fibroblasts, etc. For example, isolating adipose stem cells from the stromal vascular fraction via collagenase in culture. There are several instructional videos on the AllCells website that give even more detail on the handling of primary cells, including freezing and thawing techniques, We encourage everyone to check out the protocols available on our website. The hematopoietic system contains many different types of cells, including stem cells, differentiating cells, and fully specialized cells, all of which can be isolated and purified for your primary use at AllCells. After cell isolation, other cell types can be generated via further tissue culture and differentiation, often with cytokine, growth hormone, and peptide cocktails. This includes several of the specialized cells of the immune system. For example, specialized compounds can be used to stimulate T helper cells into activated states, and specific formulations of media are used to generate M1, the anti-inflammatory, or M2, the pro-inflammatory macrophage. Aside from the hematopoietic system, other cell types and lineages are achieved through culture and differentiation. This includes tissue derived from mesenchymal lineages derived from the stromal portion of the bone marrow. These cells are multipotent, grow in adherent cultures, and often require more than just chemical cues and treatment to achieve certain phenotypes. For example, in generating muscle cells, and in particular cardiomyocytes from mesenchymal stem cells, additional mechanical cues are required to achieve the correct tissue morphology. This is often performed in a bioreactor setup with a tissue scaffold that can provide structural support for differentiation. Also pictured is a perfusion culture used in tissue engineering. Constant media replenishment is achieved, as well as specific shearing forces, which helps the growing tissue achieve the correct morphology and physical properties. In generating the different lineages of primary cells, a number of assays and tools are available to characterize the phenotype. Multicolor flow cytometry is a common technique in which cell lines are phenotyped. This involves detecting fluorophore-conjugated monoclonal antibodies that bind to extracellular or intracellular proteins, a common example being the cluster of differentiation molecules. The basic output from flow cytometry allows the scientist to visualize and quantify differences between cells based on protein expression, size, and granularity. In addition, with certain types of cytometers, cell sorting can be performed in which subpopulations are isolated from the parent population for subsequent culture or processing. Additionally, there are a multitude of diverse flow cytometry-based assays that can be used to determine the density of a certain membrane receptor or the cell cycle phase of a certain rare subpopulation. Another method for characterizing cells, and particularly those of the hematopoietic system, is via multiplex Luminex. Somewhat similar in principle to flow cytometry, in Luminex, fluorophore-conjugated microbeads bind to cytokines and proteins expressed by cells in culture. The fluorescence is converted to a digital signal and a standard curve is applied to get an output in terms of analyte concentration. This allows for high sensitivity detection of various molecules being secreted by the cells. In the example below, the investigators were performing cytokine profiling on various meningococcal vaccines. The color spectrum indicates an upregulation or downregulation of concentration when compared to a standard. Additionally, Luminex can be done in a variety of assays and not just for immunological purposes. There are also Luminex setups for detection of nucleic acid receptor ligand binding and enzymatic assays as well. In conclusion, primary cells have been demonstrated to be of great value in medical research because they provide a more accurate in vivo-like systems than cell lines, which can contain genetic aberrations and are not consistently characterized in academic research papers. Additionally, with some expertise like at AllCells, various primary cells are easily sourced from the hematopoietic system through immunomagnetic separation and whole tissue isolation. Primary cells have also been clearly described in interdisciplinary research in biomarker discovery, drug development and screening, immunology, and tissue engineering. Finally, after isolation, primary cells can be cultured, differentiated, and characterized through many different assays depending on your downstream application. And with that, I'll hand it back over to Afshin for the Q&A portion of today's program.
Thank you, Erin, for the informative presentation. We constantly receive a broad spectrum of questions when it comes to primary cells, so it's great for us to have this as a resource. Before we jump into the Q&A session, I wanted to point out a few things to our audience. Today's webinar will be posted on our website at www.allcells.com/support/webinars. Give us about a week to post it on there, and we'll also send out a message to those attendees once it's posted. We encourage you to discuss with us any project needs you may have related to all aspects of primary cells. Using our webpage listed on the screen, www.allcells.com/technical support. is the best way to reach us directly through the site, and we can address any offline questions you may have. We will now begin answering any questions in regards to today's webinar, anything related to primary cells or in general. As a reminder, please enter your questions in the chat prompt to the right of your screen. Let's go to some of our first questions now. The first question we have here is, what are the minimum specifications for primary cells products from AllCells?
So that's a good question, since many researchers are worried about quality of cells being sent overnight. We have a minimum viability of 90%. So all of our products must be 90% viable before, upon receipt. We also have purity thresholds. So for most of our cells, our magnetically isolated primary cells, we require them to be 90% pure. Finally, we also do a quality control in terms of sterility. So we make sure that the cells are clear of biological and viral contamination before we send them.
Great, thanks, Erin. Let's go to our next question here in regards to primary cells versus cell lines. This is something we covered earlier in the presentation, but the specific question is, are there any research scenarios that would require and or benefit from using cell lines over primary cells?
Another good question. The answer to that is yes. As I mentioned earlier in the presentation, there are some applications that would actually, that cell lines would be preferable to use if you're doing large-scale production of antibodies, for example, in a manufacturing application. Also, if you're at the early stages of an experiment and you are using untested reagents, or you're just kind of in the exploratory phase and you need a high volume of cells to work with, and you don't want to waste a more valuable resource such as primary cells, you may want to start out in a cell line model. I would just always recommend, and many publications are requiring, that you actually can reproduce the results that you see in your cell line model in a primary model. So after you've verified all of your reagents that work and you've created a model that works, to then be able to reproduce that in the primary cell line would be extremely important for the majority of applications.
And we have another question from the audience here. It says, and this is kind of related to, I think, some of our BioTube protocols that we had posted before, but what do you recommend when thawing primary cells for treatment? Is there a protocol or time that you recommend placing the cells in culture before treating?
That's an excellent question. In terms of thawing, a critical point that's very different between cell lines and primary cells is the thawing needs to be a lot gentler. It's easy to do, it just needs to be a lot gentler. You have to drop-wise place medium in when you wash the cells out of the freezing medium, and we have detailed instructions for that on our website on how to do that exactly correct. In terms of how long you want to keep them in culture before treating them, I always recommend to try to let them rest overnight, as I would for a cell line after thaw, to basically allow all the metabolic processes in the cells that are normally on to kind of recuperate after the freeze.
Great, thanks, Erin. And we have another question here from our audience regarding some of our services that we provide here at AllCells. Specifically, the question comes from researchers. Says, if I come across a specific patient specimen, let's say leukemia or lymphoma patient, and would like to immortalize that specimen, does AllCells provide such service?
I have to say that is probably not something we could do here for you, but if you can definitely reach out to my email on the screen, and I can talk to you one-on-one to see if that's a possibility. Here at AllCells, we do have the capability of doing lots of assays. However, we're not set up to actually immortalize cells with the proper quality controls.
And if there, let's see, looking to see if there's any further questions. And at this point, looks like we've answered all of your questions for today. Let me see, there might be one more. And we have one more question here regarding fresh and frozen cells from the same donor. Have you noticed differences in response between fresh and frozen cells from the same donor?
So that's a question we get frequently here. Although we haven't done that direct experiment in-house, we do have a lot of customers who've done those experiments, and they report back that the results are similar. The literature is consistent with that. If you look up, people have compared fresh and frozen PBMNCs, for example, and example for a standard like response to a strong stimuli such as lipopolysaccharide, there's a definite there's recognized that the response may be dulled, but that the relative difference between donors is maintained. So I hope that helps answer your question. But in terms of functionality, I would say that the functionality is very similar. And anecdotally, all of our customers who have done that kind of experimentation have gone back and just used the frozen cells because they responded well enough.
Great, thanks, Erin. And I think that concludes all of our questions for now. Once again, if you have any other further questions or would like to contact us offline, please use the information on the screen. We'll provide this information in the slides as well that we post eventually. That concludes our webinar for the day. On behalf of Erin and the AllCells team, I'd like to thank you for your time and your interest. We look forward to hosting more educational presentations from AllCells in the future. Thank you.